Prenatal Genetic Testing by Gabby Hermes and Heather Motiff. Prenatal testing is used to look at a baby's DNA before they're born. So what is DNA? DNA is the instructions for life. Our DNA gets condensed and packaged into 23 pairs of chromosomes when cells are replicating. We get one set of chromosomes from mom and one set from dad. These chromosomes hold our genes, and genes tell our bodies how to develop and function. A karyotype is a visual representation of what all of our chromosomes look like. Prenatal testing is very important and helpful in detecting chromosome abnormalities found in the fetus. Although there are some lesser used prenatal diagnostic techniques, chromosome analysis is the primary test offered by doctors and can be supplemented by additional analysis if necessary. Prenatal testing allows parents to be more informed about their children's possible health needs and plan for any additional care or assistance the child may need after birth. Why is prenatal testing done? During pregnancy, if routine screens of the blood and ultrasound show abnormalities, or if there is a family history of any chromosomal abnormalities or genetic disorders, the doctor may order one of these tests to provide a specific diagnosis. There are two common techniques to obtain samples of the fetal DNA. Chorionic villus sampling is used earlier in pregnancy from around 10 to 12 weeks. Using an ultrasound as a guide, the doctor inserts a tube to direct the needle through the cervix and into the placenta to obtain the sample of the chorionic villi. Once in the lab, the fetal cells are separated from the maternal cells to be analyzed. Amniocentesis is used later in pregnancy, around 15 to 20 weeks. An ultrasound is also used to guide the needle through the abdomen into the uterus to obtain the sample of amniotic fluid from the amniotic sac. Both tests have a small risk of miscarriage. CPS has a 0.5 to 1% risk, and amniocentesis has a 0.1 to 0.3% risk. Doctors are looking for chromosomal abnormalities, which could be missing or extra chromosomes or pieces missing or inserted, changes in the genes, which can indicate a genetic disease, and neural tube defects, which are problems with the brain, spine, or spinal cord.
In the lab, there are several steps taken to get a picture of the chromosomes to analyze. Chromosomes can only be seen during mitosis, which is when cells are dividing. First, they separate cells that are dividing from those that are not and grow the dividing cells in culture. Next, they stop the cell division process in metaphase using a chemical treatment. This phase of mitosis is when DNA is the most compacted into chromosomes. Then, they treat the cells with a solution that causes them to burst and release the chromosomes onto a slide. They wash away all the other cell debris. A dye is used that stains regions with lots of DNA bases, adenine and thymine. This region has less genes and incorporates more of the dye than the regions containing the genes. This creates a specific light and dark pattern for each chromosome. After staining, a picture is taken under a microscope so the chromosomes can be arranged by size and counted. To analyze chromosomes, they first need to be sorted. They compare length, position of centromere, where the pairs join, and location of the bands, then line them up in order. Sometimes an entire chromosome is missing or an extra one is present. This is called aneuploidy and often comes from the mother's gametes. Most monosomies and trisomies are fatal. This happens because of something called non-disjunction during one of the two phases of meiosis, which is when gametes are replicating. The duplicated chromosome or sister chromatids fail to separate, which results in a gamete with no copies and one with 2 copies. When this gamete is fertilized, you end up with a missing or extra chromosome. In addition to counting the chromosomes, they look for structural changes of each one to make sure there are no deletions, duplications, inversions, substitution insertions, or translocations. Karyotype analysis can detect the larger chromosomal abnormalities. Amniocentesis and CVS testing also routinely screen for neural tube defects. For example, spina bifida, one of the most common, is when the spine doesn't close completely and the spinal cord is left exposed. They look for levels of a protein in the amniotic fluid called alpha-fetoprotein that increases if there's a neural tube defect. Molecular genetic studies are done if there is a concern for a specific genetic mutation that is not large enough to detect through chromosome analysis. If there's a family history or parents are carriers of a disease, these diagnostic tests can detect these abnormalities during pregnancy. Prenatal testing can provide valuable information to parents about the health and future of their baby. Meeting with your doctor or making an appointment with a genetic counselor will give you more information about the testing to determine if it is right for your family. Newer testing methods have recently become available that are less invasive. However, this simple blood test cannot determine with certainty if your baby has one of these defects. It only indicates an increased risk, and further diagnostic testing would still be needed. A new blood test developed by a team at the Beijing Genomics Institute in China successfully sequenced the genomes of 2 fetuses. This research demonstrates the possibility of comprehensive, non-invasive prenatal testing in the near future.